there, but yeah, it should be live in a second.
There we go. Okay, me again. We're gonna get going here on the next talk. So this is Lalamond and Renaissance BioScience. So I'm just going to introduce Jessica Swanson. She's the lead development scientist and beverage unit manager at Renaissance BioScience. She's got a Bachelor of Science from the University of New Brunswick and is one of the founding employees of Renaissance BioScience and currently holds the title of lead development scientist and beverage unit manager. Jessica and her team are responsible for beverage yeast development and application testing. She has a passion for craft beverages and collaborating with breweries, wineries, and distilleries to evaluate new and novel yeast strains. Welcome, Jessica.
Hi guys, so yeah, I'm here to talk to you today about hybridization, which I believe is the future of brewing yeast. And I don't know if Julia actually remembers, but maybe like 7 or 8 years ago, the very first brewing hybrid we ever made, she did a trial of in the Steamworks pilot facility. It's pretty funky and wild. I think we've come a very long time since then, but I think it's a very small world. So who we are, we're a Vancouver-based company, and the division that I manage is Beverage Yeast, and we specialize in hybridization and classical breeding techniques. And in 2020, we licensed our portfolio of craft beverage yeast to Lalamond. So, and subsequently, they've been releasing a few of our brewing strains under the Lalbrew banner. So I thought it would be nice to kind of start the talk off by going over some of the different development technologies that exist in the industry for creating new strains, starting with GM techniques. And this is where you take DNA outside of a cell, manipulate it, and then insert it into a brewing strain. So it could come from other yeast, a wine strain, or a wild strain, or bacteria. There's a few products on the market currently that were made with this technology. Sourvizier from Lalamond, which was created from their Mascoma division. It's a brewing strain that's able to produce lactic acid. And then Berkeley Yeast and Omega Yeast both have thiol creating strains, so they're able to biotransform hops. We then get into selective breeding and hybridization, which is what we specialize in. And this is where you take 2 yeast spores, breed them together, and the resultant daughter strain is a 50-50 mix of the 2 genetic contributions of the parental strains. We've recently released Farmhouse and Nova Lager with Lalamond that were both made in this fashion. If you're familiar with any wine strains, there's also Anchor Enology, who has been doing this for decades in the wine space. They do like the Vin 7 and Vin 2000 if you've ever worked in a winery or made cider with those. We then get into adaptive evolution and bioprospecting. And I would say probably 99% of brewing strains that are currently on the market were developed in this fashion, where brewers over the last, say, 100, 150 years, you've been adaptively evolving yeast to work in your breweries. So that's where the adaptive evolution portion comes in. And then strain collections and other yeast companies came in and did the bioprospecting and started purifying these and banking them and selling them. So hybrids are already widely used in fermentation. We have 8 different species of Saccharomyces strains, and then various hybrids of those Saccharomyces species. So it's not just Saccharomyces cerevisiae. There's also Kvetzii, Eubayanus, and Uverum that are widely used in fermentation, and multiple hybrids of each of those. So now that we've been able to get into sequencing and understanding the genomes of these yeast, some yeasts that were thought that were maybe just Saccharomyces cerevisiae previously, we've actually learned are Saccharomyces and Kvetzii hybrids. There's been some recently found in Trappist beers. There's obviously the famous Pasteurianus, so lager strains. It's actually not a strain of Saccharomyces, it is a hybrid of Eubayanus and cerevisiae. And then Bayanus strains, which are very popular in white wine making at cold temperatures. And this one has actually gone through multiple hybridization events throughout its lineage, so it's quite cold tolerant because it has the U. bayanus, U. verum, and then Cerevisiae. So even though you may not know it, you've already been using hybrids. So I thought it'd be nice to show you some of, in practical terms, how we actually breed yeast to kind of demystify it, show you some of the equipment that we use. And it was really nice having the hop talk just before here, because obviously all the new hops that are on the market came from breeding programs. So looking at new flavors and aromas. Barley has also gone through a lot of breeding over the last few decades to increase yield, pest resistance, and up the protein levels. And of course, dogs. It's pretty obvious. Dogs, they're the same species, but like wildly different phenotypes. And that's because of adaptive evolution and breeding programs. So why do brewers need to be interested in breeding brewers' yeast? So some of the technical traits that we're able to look at and combine and manipulate. So attenuation, looking at DP3 consumption, flocculation, increasing or decreasing, kinetics, speeding up a slow strain or slowing down a strain. This plays into flavor and aroma a little bit. So the strains that take off really quickly and ferment fast typically make more aroma. So if you want something a little more neutral, you're looking at something that has a little bit more moderate fermentation profile. Flavor and aroma, different ester profiles and things of that nature. Stress tolerance is one that we've been playing around with a lot lately, looking at alcohol tolerances, temperature tolerances, osmo tolerances. We can do sulfur reduction, so there's certain medias that we can grow yeast on that will indicate color change when sulfur is produced. So we can look at the daughter cells that have the least color change on the media and just carry those forward every generation until the point where you find a yeast that just doesn't have any sulfur production anymore. Temperature profiles. This is one that we're playing with right now too, looking at breeding strains that are either thermotolerant, that like a Kveik strain that can ferment really warm, and potentially hybridizing that with lager strains or cold tolerant strains on the other end. So you can have one yeast that can kind of span the temperature range. VDKs as well, reducing diacetyl and VDKs in strains. So VDKs are produced via the catabolism of valine and isoleucine, two amino acids. Certain strains just don't prefer to uptake them from the wort. They would rather make them themselves. And when they make these amino acids, you make the alpha-acetolactate and subsequently diacetyl in your fermentations. So strains will do this at varying degrees. So we can take a strain that likes to consume these from the wort and breed it to a brewing strain that maybe prefers to metabolize them themselves. And then lastly, enzyme activity. A lot of enzymes for biotransformation or glucoamylases are found in wild yeast or non-brewing strains, so wine strains. So looking at breeding those into a brewing background. So how it works. So this is a micromanipulator. And Zachary Turgeron, our lead research scientist there. So yeast can reproduce in two different ways. One, budding, which is what you guys are doing in your fermenters for that first 24 hours. Basically, when a yeast is in an environment that is really happy and healthy, it'll just produce a genetic clone of itself because it's doing really well, and it knows that its daughter cell is also going to do well. So it'll just produce a clone of itself and on its way. But if you put yeast in its stressful environment and it's not doing well, it'll look to mix its genetic material with another yeast with the hopes that its daughter cell will have more stress-tolerant genes to carry forward. And that's basically what we do. So we can take two yeast, we grow them in a special media with really low nitrogen and an unfermentable carbon source, and it stresses the yeast out and it turns on all those genes required for staying alive, and it'll sporulate. We then take those spores and hybridize them together, and you get a hybrid that's a 50-50 mix of the two parental strains. This is a picture of the actual microscope that we use. So you literally just sporulate the yeast and pick a single spore up using a very fine needle under the microscope. The microscope has a nice camera on it. And there's 2 little joysticks that the scientist actually uses, and they pick up a single yeast cell and just put them beside each other on an agar plate. And if the cells are of opposite mating type, they'll just naturally mate themselves. So I know it sounds pretty straightforward, and it actually is. It comes into the whole phenotypic selection and everything we do after that's a little bit more sophisticated. So this is actually a picture of underneath that microscope what it looks like. So we take the spore pair and put them beside each other on the agar plate. They then start schmooing, and that's the actual real scientific term. They release pheromones, and if they're of opposite mating types, they'll start migrating towards each other on the agar plate, and then they'll fuse their membranes and start sharing a cytoplasm. And then if that goes well, they'll share genetic material. Their nucleus will actually migrate towards each other, and then they start sharing genetic material, and that's where you get your hybrid. And the hybrid is a 50-50 mix of these 2 spores that you started with. So it's simple but elegant. It's a little bit more than just hybridization what we do. And I'll use the example of incorporating a biotransformation enzyme into a yeast. So if it would just be a 50-50 mix of potentially a wild strain or a wine strain, which is a poor brewing strain, and a brewing strain. So it would be kind of 50-50 and not necessarily the best quality strain. So we then take that and back cross it to the original brewing strain. And every time we do that, it cuts the genetic contribution of the poor brewing strain by half. So by the 6th, 7th, 8th, you basically only have the technical trait that you've been carrying forward. So at every step, we're making sure it has that beta lyase gene, it has that STA1. We carry that forward, but the poor brewing wine strain or wild strain gets diluted each time. We also do intensive phenotypic characterization, so we want to make sure that all of these daughter cells and all the spores that we're generating have the characteristics that are required to brew different styles of beer. So we want to make sure that they're meeting up with industry expectations of that style. So we don't want to put out an IPA strain that doesn't consume DP3s and produces, a 65% attenuation. So we do full volatile component screening of all of these. We do 20 different esters and fusels at every stage and make sure that they're not throwing any crazy aromas. We also look at sugar consumption, flocculation, viability, and just metabolite evolution over time to see the speed of the yeast. And again, make sure it's not throwing a bunch of acetic acid or anything crazy. We found it's also very important to do sensory and benching of all of these. Your nose is actually much more capable of measuring things in the parts per trillion, which can be really hard to measure analytically. So the big one here is sulfur components. It's almost impossible to measure them in the lab in an accurate way when they're in really small amounts. So we want to make sure we're doing benching and also make sure that we're meeting industry expectations of the style. So a Saison, we want to make sure it's nice and fruity and phenolic. So now that we kind of went over a little bit about how we breed and that, I thought it would be really good to show some actual examples of things that we're working on or have worked on and that Lallemand has released. So the first release we've done with them is a non-diastatic Saison. So we wanted to make sure we ended up with a product that was non-diastatic, but still had that amazing Saison flavor and aroma, and that wasn't just a blend of yeast. It actually is a hybrid that's 50-50 genetic contribution. So you can repitch this as much as you want, and it's gonna give you consistent results every time. So another thing that we try and breed into all of our strains is a non-H2S technology. And we start with an Italian wine strain that was isolated in a vineyard by UC Davis in bioprospecting. They have a really wonderful bioprospecting program there where they send out master's students all around the world in these different vineyards, and they bring back yeast to the lab, and then they screen them all for different traits. And they found this one really interesting Italian wine strain that doesn't produce any H2S. We start with that, and we bred it to a very high attenuating strain that we bioprospected in Vancouver. So we ended up with a hybrid that was non-H2S, a high attenuator, but didn't have any of that Saison flavor and aroma that you're looking for. So we then back-crossed it to a French Saison and carried forward only the daughter cells that didn't contain the STA1 gene, but also had high attenuation because you also don't want a sweet Saison. So it consumes all of the maltose, maltotriose. It just doesn't touch those dextrins. And then the resultant yeast is non-diastatic, non-H2S, and has a nice Saison flavor and aroma. I threw this in here because I just personally think this is super cool. We, as a proof of concept, we're playing around with, if you do adaptive evolution with certain compounds of the yeast, so you grow them generationally over and over and over in the presence of certain compounds, it'll actually work out, kind of like working out a muscle, certain pathways in the yeast. And this is one we did to boost the phenylethyl acetate of the yeast, and which is a rose and floral aroma. We gave this, it produced 5 times the level of the starting yeast. And we gave this to a local brewing partner, and their sensory panel, without any prior knowledge of what they were tasting, were actually able to pick out, and they all reported that it had a rose aroma. So I just think this is kind of a cool proof of concept of you can really play around with flavor modulation as well through adaptive evolution. Another big thing that's, I know, the talk of the town in the industry is maltose negative hybrids for low and no alcohol beers. A lot of these current products that are on the market are non-Saccharomyces species, so they're wild yeast that aren't accustomed to the brewing environment. So we wanted to try and breed an actual brewing strain that doesn't consume maltose, which is a fairly hard task because that's what they've been designed by brewers to do. So some of the things we looked to improve upon of the non-kind of Saccharomyces cerevisiae species, we obviously wanted it to only consume the simple sugars in the fermentation, so glucose and fructose and sucrose, which typically in a normal wort, 10 to 15% of the sugars. And with that only 10 to 15% of sugar, it'll produce less than 0.5% ABV. We wanted to make sure that they had a beer-like flavor and aroma. A lot of these maltose-negative strains that are like Ludwigi or Lachantia don't necessarily make a beer that tastes like beer. They don't have that characteristic ethyl acetate and isoamyl acetate. We also wanted to make sure that they require no special treatment from brewers. So a lot of these strains also don't flocculate very well. They're not really repitchable. They don't propagate very well. And by choosing the brewing stain background that we're hybridizing to the maltose negative strain, we can also do different styles of low alcohol strains. So for instance, choosing a wheat beer strain that's PUF positive, we can make a stylistic low alcohol strain, maybe incorporating in some biotransformation enzymes, and also cold tolerance for low ABV lager production. One of the other interesting things that you get along with incorporating the brewing background is that a lot of these non-Saccharomyces strains aren't accustomed to fermenting in wort, so they don't consume a lot of the aldehydes that are in wort, and you get a lot of those aldehydes carrying forward into your beer, and that's what creates a lot of those papery, warty off aromas. So we are theorizing that by breeding to a brewing strain that's been evolved to work in wort, we would get a higher proportion of these aldehydes being converted into their corresponding alcohols. And then, of course, hop tolerance. A lot of strains that don't come from a brewing environment aren't very hop tolerant. So again, we started with our Italian non-H2S wine strain, and we bred it to a maltose negative strain it's a non-brewing strain that we had in our culture collection. So we could have potentially released that product as is, but it didn't have all the characteristics that we were looking for because it doesn't come from a brewing background. We then took that hybrid and hybridized it to our brewing strain, which in our culture collection, we found a very low attenuating strain that had very high aroma. I believe it's an English strain that we have. And the resultant yeast had kind of all the characteristics we were looking for. It was maltose negative, it had non-H2S, which can be an issue in some of these low OG brews because they don't have a lot of fan. They're pretty lean, so they can really start throwing a lot of sulfur. It had the beer-like flavor and aroma that was coming in from that brewing background. And then also reduces staling aldehydes, again, because it's coming from a brewing strain that's accustomed to fermenting in wort and those aldehydes. So looking at in a typical 5% beer, our ale control produced 5% alcohol, whereas we used Saccharomyces codes ludwigi as the control. These are wildly used in industrial brewing, producing low alcohol beers, but they're non-Saccharomyces strains. This is our hybrid, the MN229, and you can see that it produced, in a typical wort, produced less than 1% alcohol. And that's because it didn't touch any of the maltotriose or maltose in the beer. So you obviously would want to do recipe development on this. You're not going to put out a beer that has this much sugar in it, but it's just illustrating that in a typical ale strain or in a typical beer, they don't consume any. But it will consume, obviously, the simple sugars, so glucose and fructose. So the only alcohol production in the fermentation is going to come from the small amount of glucose and fructose that are in your wort. And in this instance, it was around, I think, 17 grams per liter. And then also looking at ester profiles. So one of the things we were hoping of in breeding it back to that brewing strain is that we would get more beer-like esters in comparison to some of the wild strains that don't come from brewing backgrounds. And that's exactly what we saw. You can see that our hybrid produced overall more esters, and particularly isoamyl acetate and a little bit of ethyl acetate, but fusel alcohols remained consistent. And aldehydes. So this is the one that we're really hoping we can make an impact on. The way we started this project, I went out and purchased as many low alcohol beers as I could get my hands on, and we ran them through our sensory program. And I thought it was going to be and we put them through full GC paneling and HPLC paneling as well, so I could see all the analytics of the beer. And looking at the analytics, I was predicting that the beers that had the most ester and aroma were going to be rated the highest, but in reality, it was the beers that had the least amount of off aromas. So if you were to take the beers that had the least amount of off aromas and put those together, maybe it would be the ones that had the highest esters that would be rated the best. But looking at everything, it's the lower the grainy, warty off aromas, the better the beers were rated. And our maltose negative brewing strain coming from that brewing background, it did and does have a higher proportion of aldehyde reduction. So the other maltose negative strains that don't come from a brewing background only consumed a very small portion, I think 1.22–1.7%, whereas our strain was getting those benefits of being hybridized to a brewing strain that has been evolved in wort and in breweries. So you get a much higher utilization of those aldehydes. It's converting them into the corresponding alcohols. This is one that we've recently launched with Lallemand. We've recreated the original hybridization event that occurred in a brewer's fermentation, I think in like the 1600s. So we've recreated a lager hybrid. So there's basically two lager strains on the market or in the industry. One being Group 1 saws, It's a triploid, so it has 2 copies of the Eubayanus genome and 1 copy of ale. This is like Carlsbergensis, if anyone's used that strain. I'd say the majority of brewing strains are group 2. This is your diamond lager yeast, your 3470, and they are 50-50, so they have 2 copies of Eubayanus and 2 copies of the ale parent. Recent genome sequencing has given us a little bit more information on where this hybrid potentially was made. And they think that it actually was made in a brewer's fermenter because the ale portion of it more closely relates to domesticated ale strains rather than a wild strain. So somehow a Eubayanus strain made its way into a brewer's fermentation, made this natural hybrid, and because it fermented so well and produced such a good quality beer, the brewer carried it forward over and over and over until someone came along in a culture collection and snatched it up and started selling it. So there's 2 ways of potentially remaking this hybrid. I think in the last 5 years, this Eubayanus here, this wild Eubayanus, was bioprospected on a tree in Patagonia, of all places. But the problem with it, if we were just to take this and start hybridizing it, is that it hasn't been domesticated by brewers. So it doesn't consume DP3s, so it doesn't attenuate very well. It leaves behind all the maltotriose, and it's also phenolic. So no one really wants a sweet phenolic lager. Maybe someone does, but I would say not most people. So what we did is we actually removed the subgenome from this lager strain. And you guys, brewers, over the last 400 years, have domesticated this through adaptive evolution, only carrying forward the good attenuators and the ones that don't produce a phenolic beer. You adaptively evolved the subgenome in the hybrid, and that's what we use for breeding. So we removed the subgenome and we hybridized it to various ale strains. And you can really start making some cool combinations here depending on what ale strain you choose as the parental contribution. So I like to think of this as apples and bananas. Ales are apples. There's a huge variety. You can breed them very easily. You can think there's culinary apples, there's cider apples, there's different flavor profiles. Those are the ales. But the lager is the banana. It's a monoclonal culture, and there's one banana. But potentially, if someone could start breeding bananas, you could do different flavors and aromas, maybe bananas that don't bruise anymore. There's lots of different options there. And so lager, think of lager as banana. Next time you're eating a banana, you can put that in your mind. For the yeast nerds out there that want to learn more of how we actually do this, we published 2 papers that get in depth on the genetics and the techniques. So the first being a very genetics heavy paper, and you can hit that QR code and it's open access, you can read it. And then our brewer, who is now at Faculty Brewing, Ryan Juergens, who's sitting over there. Thanks, Ryan. He took it and made a more brewing focus that's application heavy, and we published that with Master Brewers. So that one's probably a little more interesting for you guys. Now looking at, this is from that paper, we want to see temperature tolerances to see, do these new hybrids, do they more closely resemble lagers or ales? So we did spot plates. Lagers will not grow at 37, which is what you can see. Ales won't grow at 7 degrees. They're not cold tolerant. They don't have those genes from Eubayanus. But our hybrids were able to span both temperatures, and they will grow at 37 or 7. Looking at ester profiles and fusel profiles, our hybrids come in kind of right in the middle. They're a little bit more than traditional lager, but quite a bit less than an ale, cold tolerant ale. Same thing with the fusels. Also looking at sugar consumption at a cool temperature. So these were in a 15 Plato fermentation at 12 degrees. You can see that our hybrid in purple here ferments even faster than the parental lager strains. And that's due to, we believe, there's a theory that's pretty well known called hybrid vigor, where incorporating two new unique genetic backgrounds, you get higher diversity of stress tolerance genes, and that's exactly what we saw. Our strains were quite a bit more stress tolerant. When you start doing these breedings as well, you start with looking at what are the differences between the two yeasts that I'm going to be breeding, and what benefits can we get via breeding them. So another main difference between lagers and ales is lagers produce more diacetyl, and that's because they prefer, even if the wort has tons of amino acids, they prefer to catabolize two amino acids themselves rather than uptaking them from the wort. That's isoleucine and valine, and when the yeast makes these, that's where you get the alpha-acetolactate and subsequent diacetyl. So we are theorizing potentially by breeding to an ale strain that will readily uptake these from the wort. They don't make them themselves, and they make less diacetyl. So we're hoping that we could produce a low diacetyl strain as well. So this is a fermentation, again, a 15 Plato in 12 degrees. And you can see up here, these are the lager strains. So even though there's tons of isoleucine left, they stopped fermenting probably after day 3 and just started making their own. Whereas the ale consumed probably almost 100%, and they didn't. And then our strains subsequently consumed much more. And same thing with the valine. Even though there was lots left, the lager just stopped consuming it, whereas the ale consumed around 90%, and our strain consumed 70%, our lager hybrid. But did that translate to diacetyl production? And yes, it did. So you can see the lager and ale controls produce up to 2,000 ppbs of diacetyl at mid-fermentation. Ours had a much smaller peak because, again, it wasn't catabolizing or making those amino acids themselves. It was happily uptaking them from the wort. So we produced much less diacetyl in the end fermentation. So one kind of last example here. This is one that we've been working on lately of, can we get a single yeast that will ferment lager temperatures all the way up to Kveik temperatures? Can we make a hybrid you can have one yeast that'll do everything in the brewery? So looking at the lager strain in orange, we did fermentations at 35 all the way down to 10 and looked at residual sugar and all the metabolites and everything. It's a lot of data, so I'm only showing the sugar here. But lagers were able to consume sugars across the board. Lagers actually, they will brew beer at warmer temperatures and consume sugar. They just throw a ton of sulfur and a ton of diacetyl, so they just don't make a very good beer. And that's exactly what we saw. Anything over 18 degrees, they just started throwing a lot of aroma. So it fermented the beer and dried it, but just made a really poor quality beer. The Kveik strains worked really well at the higher temperatures, but anything under kind of room temperature left a pretty significant amount of residual sugars. And they also produced a lot of acetic acid. So they're kind of vinegary and a little off. I know there's a lot of different Kveik strains out there. This is just the one that we chose. And then our hybrid, the NHL 27.7, was able to dry the fermentations across the board like the lager strain, but in the difference, it produced a nice clean profile. It didn't throw any odd off aromas, no diacetyl or sulfur aromas. So So it's very clean and nice across the board. So thank you very much. So our commercial partner is Lallemand. I think they're here with AB Vickers. We're at the AB Vickers booth if you want to go and ask them, chat about that. And Brew Culture is serving a beer made by Four Winds with the Nova Lager hybrid. If you want to, I think it's American Light Lager. If you want to go and chat with them about it and try the beer, I'm sure that they'll be more than happy to. That's it.
If anybody has any questions, I can run up there with the mic. Does anybody? Yeah. Sure.
Thanks. Jessica, I'm curious on the last set of data you had there. What gravity were you fermenting at?
I believe this is 15.
Okay, thanks.
Any other questions?
Thanks so much for this. With the low alcohol ferments, you were targeting higher ethyl acetate and isoamyl acetate. I'm just kind of wondering, like, what levels you're getting. Is it similar to like an English ale, so it's more natural? I'm assuming not so Belgian-y.
No, and even, I'll go back to, oh, no. So you can see, even because you're only getting ester and fusel production from that small amount of sugar that you're consuming, so it'll never be in a high amount. It's just because there's not enough carbon going through the metabolic pathway. So it's only, 2.5 total esters, which a typical beer, typical lager is gonna have 30, 40. So you can see that just inherently any yeast that you put in one of these low gravity worts, it's gonna be low. So when I say high esters for a low alcohol, it's not gonna be something that's, it's still gonna be on the neutral side, but just tastes more beer-like than some of the non-Saccharomyces species.
Oh, you're gonna have to go all the way over. You too?
Yeah.
Okay, I'll stop.
On like the next slide, there's the reduction of aldehydes. I was wondering if you had info on how much a standard ale fermentation reduces aldehydes? Or, yeah.
Like a standard ale would consume probably 80%,
Okay.
90%. That's why a typical beer isn't like really wordy and aldehyde-y, but they will go over time. And that's why a shelf life on a beer is typically 3 months, because you get the aldehydes will slowly convert over that 3 months, and that's when it starts getting that papery and cardboard aroma. But a typical ale will do, probably about 80%.
Okay,
in a standard beer, not necessarily a low alcohol wort.
Any other ones? Okay. You weren't kidding about the workout.
Curious about what your thoughts are for where you're going forward with this. If you have some different yeast strains that you're planning on going ahead with in the future to hybrid, and also as a follow-up, as a brewery, how can we work together with you to, if we have an idea for something that we would like to potentially hybrid?
Sure. So we do have our commercial partner, Lallemand, and we give them lots of fun, wacky, fun ideas. And they have to, look at it and see what the market wants and what they're going to be releasing. So we work in partnership with them. I know biotransformation is something that's big in the market right now, the low alcohol as well, the widening the temperature ales for smaller breweries that might just want to have one strain that they can do everything with. So I know those are definitely top of mind. Getting into some of the more wackier, fun ideas. I want to do a proof, we haven't done it yet, but I would love to do a proof of concept of recreating this. but incorporating, instead of just doing Cerevisiae, so an ale strain, potentially doing like a Kvedzi or one of the other Saccharomyces species and doing like a triple hybrid. So potentially getting some like really interesting new novel flavors and aromas. And so I know lagers have just kind of always been, you use this one yeast and it has to be a clean profile, but I think there's also room out there to kind of start doing some more funky, cool stuff as well.
Any other questions? Oh, okay.
Make it easy for you. On the hybrid side of things, I know you talked about standard brewing practices and repitching. We run really high generations at our brewery. What is your guys' stability like with your wonderful hybrid vigor?
Yeah, just the same as any other strain. So that is something that we do stability testing on, and we do repitching. So we do 2 types of stability testing. One being because we have to grow these up, so we look at propagation stability because they have to go through a massive amount of generational growth in order to produce an active dry yeast. So that's one type of stability we look at. And then we also look at stability in an actual beer fermentation where there's alcohol stress and a lot of other stresses on the yeast. The strains actually have more stability in an alcohol fermentation because that's selective pressure. So the yeast wants to keep all of its genes. It's not a benefit to the yeast in those alcoholic and stressful environments to start dropping genes. They usually, you see stability on the other side during propagation if there is an issue. So in an actual brewing fermentation, it's exactly the same as like a 3470 or any of these other strains. So you will see genetic drift over time, but that's just, it's a yeast, it's an organism.
Hi, you mentioned a few strains you couldn't grow up and kind of prop again, and there's some strains out there that we've started to use that it's kind of a one and done. But is that an outcome of the hybridization, and is there any plans to try and make that repitchable? Because like some strains are super cool, but if you can only use them once, it gets really expensive.
Our strains are fully repitchable, and that's the benefit because I would say the word hybrid is used in the industry in 2 ways. Some people say hybrid and they mean an actual blend of yeast. So it's 2 yeast strains that you grow up and just blend in the package. But ours is a genetic hybrid, so it's just a yeast. It's fully repitchable. It behaves like any other yeast.
Anybody else? One more. Okay.
You can stop by the AB Vickers booth. I believe that they're doing trials right now, like pre-launch trials. So they go through a pretty extensive, we don't just give them something, they're like, thanks, and like release it on the market. They go through a pretty extensive, probably 6 months to a year of giving it to brewers and doing trials and getting the data back and making sure that it's a viable product before they do like the big launch and a big batch. So I believe they're in that phase now where we're doing trials with brewers. I will say that the one thing with doing these maltose negative strains is that you have to have a pasteurizer because there's residual sugar. So that I know that excludes some people from using it and doing trials.
Anybody else? Okay. Thank you so much. That was really cool. Schmooing. That was my favorite.
Thank you very much.
I have a sweatshirt for you.